Hi everyone. Today I will start a lecture on microbiology. In microbiology, today I will talk about basic introduction of microbiology. What's the basic introduction? In basic introduction, today I would like to talk about what are the infectious agents which can cause infectious diseases. So today my main focus will be on infectious agents. So let's start. So actually infectious agent is category into different types. So this is in the picture you see there is some category of infectious agent. It depends on the smallest and smallest organism. So in that category of infectious agent there will be category number 1, that will be prions, but in that picture they will be not included, but prions, which is my major topic today. So prion is also smallest organism which can cause infection. And secondly there will be virus, and then there is a bacteria and fungal and protozoal infection and multicellular parasite infection. So in our what is the most abundant organism which can cause infection or which are widely present, that will be bacteria. So bacteria is abundantly amount of present inside on the earth. So the first one is the prions, which is the very smallest organism, smallest infectious agent you can say, which can cause infection. So actually what is the prions? Prions is the abnormal structure protein, that but in our normal protein there is also same amino acid which are present in the prion. So amino acid here and amino acid present in the prion same, but depending on their structure. So here is a beta plated structure and there is a alpha structure. So according to the structure, prions is different from our human body structure protein, but otherwise amino acid sequence will be same in normal and amino acid sequence will be same in prion. But disease happen when there is a abnormal structure protein is present, like in a amino acid present in the beta helix structure, but amino acid present in the sheeted form, or you can say alpha structure. So in the normal protein they will not cause any infection, but when that protein come in the body and they change the structure of our normal protein and they make the structure of that normal protein like that structure, then it can cause what? Infection. So in the normal protein there will be no helical structure. I will give you important why that structure is very important. So up to now you just understand that in the normal protein there is the alpha structure protein, amino acid in the alpha helical structure. But in the prions, this is a prion. So in the prions there is what? This is a beta plated structure protein. So what's the basic introduction of prion? Actually the basic introduction of prion is that prions is the smallest infectious agent that don't have DNA or RNA. It's just a protein particle with abnormal protein structure. So if they don't have DNA, how it replicates? This is the major thing which I will tell you later. The prions contain the same protein that are present in normal human body, but shape will be different. So its structure will be what? Beta plated arrangement. So this is the structure. So actually if they don't have DNA or RNA, how it replicate? Actually when it come in the cell of our human body, so actually there is a, see in that picture, actually when the DNA will be transcripted into messenger RNA, and then messenger RNA come into the cytoplasm and they convert into the translational phase. In the translational phase here some modification will be happening. When that prions enter in the cytoplasm, when they enter in the cytoplasm, that modification is going on. So during that modification, when translation will be happen, after translation modification is still going on. So prions change the structure of our normal protein and they make our normal protein same like prion. So our normal protein also same like the prion. So then it can make a infection or make a abnormal structure protein. See in the DNA when it transcripted, it convert into messenger RNA, and that messenger RNA when it come into the cytoplasm, here ribosome will be acted, and this is the amino acid which are linked together to make a protein, and this is a ribosome and this is a messenger RNA. So after this translation, that ribosome is further further present into the Golgi apparatus, then endoplasmic reticulum. So modification is going on, still protein is not properly formed, still modification is going on. During that modification, listen properly, okay, so during that modification, that prions change the structure of that. During that modification, prions act on that protein and they change the structure, and as a result there are multiple abnormal structure protein present, and that we call prions. So to that reason, even prions don't have any DNA or RNA, but how it replicate, it just change the structure, because still here proper structure is not formed. Proper structure ends at the endoplasmic reticulum or Golgi apparatus. At Golgi apparatus and endoplasmic reticulum, our normal protein get its fully structured, but during that stage they are not occupy fully structured. So that's why prions get a time or get a chance that now I have to change the structure of normal protein. So during that steps, so they can cause infection. Then proteases. Actually, what is proteases? Proteases is an enzyme that are present in every cell. So what's the structure, what's the function of that enzyme? Actually, that enzyme destroy our denatured protein. So for example, when protein work properly, then it denature. So proteases that destroy that denatured protein. So that proteases only work on alpha helical structure protein. So protease only destroy that protein which have alpha helical structure, but prions don't have alpha helical structure. Prions only have beta plated structure. So that's why protein cannot destroy prions protein. So this is the main thing here, that why proteases cannot destroy, or why beta plated resist the proteases, because prions contain alpha helical. See, these prions contain the alpha helical structure, beta plated structure. So proteases cannot act on that and cannot destroy that beta plated structure. But as compared to alpha helical, proteases easily act and can destroy denatured protein and not cause disease. So proteases will not work on that protein, normal protein. Proteases can work on that normal protein, but proteases cannot work on that prions. Why? Due to their structure. Here structure is normal, alpha helical structure, but here structure is abnormal. We call beta plated structure. So this is about prions. So prions is a very smallest infectious agent. So in my next lecture of microbiology, I will talk about further about bacteria and then virus and then parasites. So that's all for this lecture. Okay. So if you have any questions, you can ask me in comment section. Thank you so much.